Hello, everyone, and welcome to today's live broadcast, Engineering the Hematopoietic System to Treat Non-Hematological Disorders, presented by Dr. Natalia Gomez-Ospina. I'm Christy Juliff-Labrutz, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by Labrutz and sponsored by STEMCELL Technologies. For more information on our sponsor, please visit STEMCELL.com. Now let's get started. I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click the Send button. If you have any trouble seeing or hearing the presentation, click on that support tab found at the top right of your presentation window, or let us know you're having a problem by using the Ask a Question box. I'd now like to introduce our presenter, Dr. Natalia Gomez-Ospina, Assistant Professor of Pediatric Genetics and of Pediatric Stem Cell Transplantation, Stanford University. For a complete biography on Dr. Gomez-Ospina, please visit the Biography tab at the top of your screen. Dr. Gomez-Ospina, you may now begin your presentation.
So thank you, Christy, for the introduction, and thank you all for attending this webinar. Hopefully you're all here because you want to learn about how to engineer the hematopoietic system, and then you can use this to treat diseases other than blood disorders. So we'll first review the genetic modification tools that exist for hematopoietic stem cells and discuss the pros and cons of these different tools. We'll then discuss how we can use hematopoietic stem cells to treat non-hematological diseases. Specifically, we'll review studies for efficacy and safety for one lysosomal storage disease, and we'll wrap up with a summary to highlight the take-home messages. But first, we should probably ask why engineer the hematopoietic system. So I'm here to argue that our ability to do this precisely and well has therapeutic implications in several domains of human health. So there's the acquired diseases, and we already have a lot of great results engineering T cells and CAR T cell therapy. Of course, there's an impact on genetic diseases, blood and hematopoietic and hematological and non-hematological. But I think they could also bring solutions for aging and disease prevention. The idea here is to really leverage the fact that the hematopoietic system is perhaps the best understood system, and that it's the one that's really kind of most fulfilled its promise in regenerative medicine. Unlike any other organ system in the body, we have decades of experience isolating and manipulating the stem cells and transplanting them. So why not try to use all this towards as many diseases as we can? While I mentioned that we are talking about therapeutic implications in several domains of human health, we will focus here on the use of these genetically modified hematopoietic stem cells to treat genetic diseases. And this slide is here to convey the message that genetic diseases are an important medical problem. There are millions of people affected in the United States, hundreds of millions worldwide. Many of these affected people are children, and a good number of these children will die of a genetic disease before the age of 5. And there are very few FDA-approved therapies, but even fewer therapies that are one-time or definitive, which is what I'm really interested in developing. Now we have we have been genetically modifying hematopoietic stem cells for decades. Recombinant, replication-deficient retroviral vectors have been really the most common tool so far for permanently altering cellular DNA in these cells. And so they've been in development for decades, and there were some serious adverse effects in the early clinical studies, but this led to more research and safer vectors, and fortunately there are now viral therapies that are available for patients with a variety of genetic diseases. And so while this webinar is focused primarily on genome editing, it's important to acknowledge that the work with integrating virus is still quite relevant today for hematological and non-hematological diseases. Now, despite tremendous progress in gene addition using viruses, sometimes you really want precise genetic modifications, and this is important if you want to achieve predictable patterns of gene expression, or for targeting specific alleles. You can't do that with viruses. So genome editing, as you all know, is a mechanism to very specifically rewrite DNA sequences. And typically we think of genome editing as a tool to turn abnormal sequence, shown here as incorrect, to a normal sequence, shown here as sort of correct. But I'd like to say that, in fact, perhaps more importantly, genome editing allows us to create new sequences that sometimes can have even more therapeutic value. Now, there are several types of genome editing tools. Some are nuclease-based and some are not nuclease-based. At the core of all nuclease-based genome editing platforms is the ability to create double-stranded DNA break at a specific DNA sequence or genomic location. The differences between all these different platforms is going to be really based on the mechanism by which the different nucleases are recognizing their target sequence. So engineer nucleases like the zinc finger nucleases and tailings are these proteins that have really two parts to them. One consists of a customizable sequence-specific DNA binding domain, and then they're bound to an endonuclease, the FOC1 endonuclease, shown here with this scissors. So zinc finger nucleases on the left, their DNA binding domain consists of this array of zinc finger DNA binding domains, and for the tailings, the DNA binding domain is composed of these repeats derived from transcription activator-like factors. Both of these types of platforms require a lot of protein engineering and complex synthesis, so it has really limited their widespread adoption. But between these two platforms, ZSMs have been the most extensively developed for therapeutic purposes, and in fact were the first ones to make it to patients. An additional platform are the RNA-guided nucleases, which are derived from CRISPR-Cas system. For research and therapeutic purposes, what we use is sort of a modifier, or better said, engineered version of the system that basically has two main components: a guide RNA, sometimes abbreviated as sgRNA or gRNA, and CRISPR-associated endonuclease, which is a Cas protein. Because of these, and for research and therapy, most of the Cas that are used are Cas9 nucleases derived from strep 5 mutant Staph aureus. Now, because of the RNA-based recognition, targeting different sequences only requires changing the RNA. This is much easier, so this versatility has driven their widespread rapid adoption, not just to modify genomes for basic research and therapeutic applications. An additional platform, which is a derivation of this, is the base editors, which are shown here on the right. These guys make use of a catalytically inactive Cas9 or Cas9 to target base-modifying enzymes such as cytosine deaminase or adenosine deaminase to very specific locations. So an adenosine deaminase would turn adenine into guanine, and cytosine deaminase will convert cytosine into thymine. So this offers you a couple of possibilities in terms of modifying these specific residues within a narrow window of the RNA binding site. So there's no double-stranded break from this particular platform, which presumably makes them a little bit safer. It's likely to make a cut in a place that you didn't intend. On the other hand, you're limited to modifier seasonings, which is going to be good for a lot of things, but not good for everything. So we begin this editing in general with the nuclease-based platforms with a double-stranded break. This in the cells will trigger a couple of double-strand break repair mechanisms, either non-homologous joining, abbreviated as NHEJ, or homology-directed repair as HDR. So NHEJ on the left here typically does a good job of repairing the break, but if it's repaired properly, regenerates the target sequences, which causes subsequent iterations of break and repair, until there is a mistake that it's no longer recognized, so it gets cemented in the beginning. These mistakes tend to manifest in the form of insertions and deletions, and we tend to abbreviate this as indels. And so non-homologous joining gives rise to indels. On the right, we're showing homology-directed repair. So you can do this, the cell can do this if you provided with a DNA template, and the way that it knows to use that template is because that template has homology to the regions of DNA around the double-stranded break. This is a little bit harder to do, and then you also have to deliver the DNA template, but both repair mechanisms can happen with high efficiency if you're using optimal conditions, and we'll give you examples of this. So NHEJ, although it seems like it's good for breaking things, it actually has therapeutic application. So on the left, I'm showing you an example of indels generated by one of our therapeutic guides, and what I want to show you is that it really results in a mixed population of alleles, and this is not hard to control. It varies for each guide, and it varies per cell. So the NHEJ outcomes are harder to control, but nonetheless, you can use this for therapy. So you can use it if you use NHEJ to target a coding sequence. You can prevent protein expression, and that has therapeutic uses. You can use it to target regulatory elements, shown here in 2, like a promoter, enhancer, or microRNA. And the final effect in protein expression can be repression or activation, depending on the role of that element. Here in 3, I'm showing you, like, if you use 2 guides with this, you can actually create more precise deletions. And this, of course, can knock out a protein, but you can actually use it to restore protein expression in cases where mutations have disrupted reading frames or have created cryptic splice sites. This is also very useful for pathogenic expansions, like trinucleotide repeat expansion. And interestingly, you can also use NHEJ to restore reading frames. So if your indels are mostly 1 or 2 or 3 nucleotide insertions, then that can actually, in specific situations, restore the reading frame. So it doesn't only knock things out. Therapeutic HDR. Therapeutic HDR, again, requires a template you can make more precise changes. So number 1 here is the most commonly used approach, which is to use it to create change single nucleotide variants. It's the most efficient way of doing this. But for most genetic diseases, this is useful in diseases where there's a prevalent common pathogenic mutation. An example of this would be something like sickle cell disease. But for most of the genetic diseases, your mutations are throughout the coding sequence, so you may want to opt for number 2 here, where you can actually supplant the entire coding sequence under the control of the endogenous promoters, and this provides a single platform for all pathogenic variants. Now, nowadays there are limitations to the size of these repair templates, so it's possible that you can fit your entire cDNA here. So you can go for option 4, where you could replace 1 or several exons, and ideally you could do mutational hotspots as well. In number 3 here, I'm showing you where you can, an example of integrating sequences that contain both a promoter and a coding sequence, and these are generally best placed in genomic loci known as safe harbors. These are well-characterized regions that are, dispensable for the particular series of targeting or the organism, and we'll talk a lot more about this. HDR can be used to insert polypeptide sequences that are useful for a lot of reasons. Basic research, they're used for visualization, isolation, localization, but depending on what types of sequences you're adding, that can actually have enhanced therapeutic effects. And HDR can be used also to make very precise deletions. So in addition to producing the desired genetic mutations for genome editing to be therapeutic, these components really must be efficiently targeted to the appropriate cells in the appropriate organs, depending on the disease you're going after. So there are 2 approaches for modifications: ex vivo, which is outside of the body, and in vivo, directly into the body. In the ex vivo modification, you have to, the modifications, the cells, the target cells might be isolated and available for transplantation, and consequently, they have to be central to the disease you're going after. So for the most part, therapies that are targeting hematopoietic stem cells are done ex vivo, like as shown here on the right. There are main advantages of using an ex vivo approach is that you have control over the cells that you're targeted, which you don't if you just inject this component in vivo. And you have the ability to perform pre-transplant characterization of your gene editing outcomes, which, there's still a question of whether you have unintended genomic modifications, or it's better to have an idea of if that's happening before you transplant the cells, and also gives you the ability to thoroughly examine the degree of phenotypic correction in the cells before you transplant them. So we'll now focus on the use of CRISPR-Cas9 to modify human hematopoietic stem cells, stem and progenitor cells. I'll try to abbreviate this as HSPCs from now on. So as we briefly discussed earlier, depending on where you're intending to do therapeutic NHJ or therapeutic HDR, you must deliver 2 or 3 components in these cells. And the biochemical version of each component can vary. So for example, the Cas9 nuclease can be delivered as a plasmid, a viral genome, an mRNA, or protein. The RNA can be delivered as a plasmid, a viral genome, a dual guide system, or single guide system. And the repair template can be delivered as a plasmid, a viral genome, or even a single stranded DNA molecule. So for HSPCs, we've learned that the most efficient way of doing this is to deliver the Cas9 as a protein. The guide is a single chemically modified guide, and the donor template as a viral genome. And so where does this weird protocol come from? So the first was a seminal paper published by Eyal Hendel and Rasmus Beck while they were in the Porteous lab at Stanford. Here they show that chemically modified RNAs enhance CRISPR-Cas9 in hematopoietic stem cells, and particularly the guides that had chemical modifications that included a 2'-O-methyl, 3'-phosphorothioate worked well. And the idea that these guides show a decreased toxicity, likely because there was less activation of innate immune responses to naked RNA, and this resulted in higher efficiencies. Now, the idea of using AAV to deliver the DNA repair templates was really established by this paper from Paula Cannon's lab. Here, where they show that the AAV, they compared different serotypes, and they show that the AAV6 serotype resulted in the highest efficiency of homology-driven genome editing events. This slide shows you a flowchart of a typical experiment using human hematopoietic stem cells. It's beautifully depicted in a Nature Protocols article that was published last year. After the cells are isolated, whether from cord blood or peripheral blood or bone marrow, typically the cells are expanded. Right now we're expanding them for about 48 hours, but people are working on methods to expand them for longer periods. This usually involves the addition of molecules that will preserve stem cell potential. After expansion, the cells are modified using the Cas9 protein in the guide, delivered as ribonucleic proteins using electroporation, followed by transduction of the AAV6. After some time for expression of the therapeutic protein, then what you want to do is analyze the HRF efficiencies, other reported expression, if that's what you have. We're using genomic methods. We'll discuss droplet digital PCR for this. You also want to characterize protein expression, and this is also a good time to analyze the effects of modifications, such as effects on clonogenicity or progenitor differentiation, and using colony formation assays and HSPCs. If you're attempting to design a one-time therapy, it's important to make sure that your modified cells are still stem cells by looking at their cell renewal capacity, using long-term engraftment studies and their ability, as well as examining their ability to differentiate into multiple hematopoietic lineages, and you examine the differentiation in vivo as well. Ultimately, you want to assess the efficacy of your cells in the appropriate animal model, and what you do really depends on the disease process that you are going after. So now we'll show you examples of what these experiments look like when applied to a non-hematological multisystemic disorders such as lysosomal storage disease. So brief intro to these diseases. So these are a large group of genetic conditions that result from deficiencies in lysosomal enzymes or proteins in general. Lysosomes are important centers for catabolism within cells. They're in charge of degrading and recycling macromolecules. So you can imagine this sort of incendiary function is pretty essential for most cell types. So when there's lysosomal dysfunction, this can affect a lot of different cell types, and they manifest as problems in a lot of organ systems. Cells in the brain, however, are exclusively susceptible to lysosomal dysfunction, so this is the reason for the majority of these diseases have neurological involvement. So many of these genes involved in LSDs encode for lysosomal enzymes that are responsible for degrading the metabolites. Not all of them are enzymes. Some of them are membrane proteins. Now, Many of these enzymes are normally secreted into the extracellular space. They are modified with the mannose-6-phosphate residues, which I'm showing you here with the red blobs, and they can be internalized by any cell that has a mannose-6-phosphate receptor. So what this means is that a cell with enzymatic activity can effectively correct a cell without enzymatic activity, and this is the property that is known as cross-correction, is the basis for all the therapies that have been designed for these diseases. So a corollary to the property of cross-correction is that you can reconstitute enzymatic activity and some enzyme depot in the body, and then that should treat other organ systems that you're not directly targeting or correcting with your genome editing approaches, for example. So our goal here is to use the hematopoietic system to really cross-correct our organs. So a canonical LSD is this disease called mucopolysaccharidosis type 1, abbreviated as MPS1. It's also known as Hurler syndrome. This disease, as you can see from the table, affects multiple organs, including the brain, as I mentioned earlier. Like many genetic diseases that are due to enzymatic problems, there's degrees of symptoms depending on how much residual activity the patient has. So patients with severe disease have progressive cognitive impairment, which without treatment, it's fatal on the first decade of life. Patients that have some residual activity have less cognitive issues. At the bottom line, I'm highlighting the residual and enzymatic activity for the different group of patients. So severe patients have some right around between 0 and 0.2% of residual activity, and patients with attenuated disease have about, let's say, a median of 0.8% enzymatic activity. So you can see that a very little enzyme makes a huge difference in the natural history of this disease. And this is important to know when you're designing a therapy, because it tells you that the realistic therapeutic threshold you're trying to achieve, which means that for the disease it's not 100% or even 50%, but a few percent will make a big difference. So you might think this disease is extremely rare. Why should I care about that? It actually this is a disease that is not part of several newborn screening programs in several states across the United States. So those states are shown here in blue, including my home state of California. So this disease is the result of an absent lysosomal enzyme called iduronidase, abbreviated as IDUA. And without this enzyme, glycosaminoglycans, abbreviated as GAG, accumulate throughout the body, and this results in this progressive multisystemic disease. Patients with this disease have currently 2 treatment options. Enzyme replacement therapy, or ERT, is the intravenous delivery of a recombinant enzyme. It's expensive, requires chronic administration, doesn't treat the brain because it doesn't really cross the blood-brain barrier, and it is also not very good treating bone. The other option is allogeneic stem cell transplantation, or allo-HSCT. This has been shown to arrest and prevent cognitive decline in children, but because an allogeneic transplant involves the transfer of the immune system from one person to another, it can have significant morbidity and mortality. And so because of that, we really reserve it for children that have severe disease, that are before the age of 2 and that have normal cognitive evaluation at the time that we find them, so that the risks are worthwhile undertaking. So how does a hematopoietic stem cell transplantation work? So in this disease, it works better than ERT because, number one, it provides a constant reservoir of the enzyme. And so most importantly, because the bone marrow generates these monocytes, and after that, tissue macrophages that can migrate into recipient tissues like liver, spleen, brain, and lung to deliver your therapeutic protein, in this case, lysosomal enzyme locally. This migration is triggered in part by the condition and regimen used during the hematopoietic stem cell transplantation, but also by the disease process that causes inflammation within the organs themselves. So given some of the facts that I've given you, are what we think. So you can see that there is a very important role for these cells and the therapeutic benefit of HSCTs in treating other organs. And so From the facts that I've given you, it makes sense to propose that the treatment for this disease and even some of these other diseases can be improved by establishing an autologous transplantation of gene-edited hematopoietic stem cells. The general idea is to isolate the patient's own hematopoietic stem and progenitor cells, which are positive for this marker CD34. We modify them ex vivo. We're using CRISPR-Cas9. Instead of targeting individual mutations or even individual genes, we decided to target enzyme expression cassettes into a safe harbor, as we described earlier, and referred to as a targeted gene addition approach. The idea is that these cells are then transplanted back into patients where they will reconstitute blood and use these also macrophages that will treat other organs. Now, I wanted to talk a little bit about the safe harbor idea. So again, it's a locus that doesn't have an essential function in development or health. In this example, we use CCR5. We consider this a safe harbor because there are millions of people in the world that have deletions in this gene, particularly in the European population. More than 10% of them don't express this protein. And this observation, as a result, it was noted that patients who didn't express CCR5 are also resistant to HIV infection. So we know we don't need this. There are several advantages to using a safe harbor. One is obviously you don't have to design your therapy for every mutation because patients usually have different mutations, so you don't have to redesign the therapy every time. The second thing is that in this particular diseases, we know that you're kind of using the blood to express an enzyme, and normal levels of expression are not sufficient. So you want to drive supra-endogenous expression, and you can only do that by using this genetic approach. The other thing is you have a clean sequence that you can further engineer to enhance therapeutic benefit, and you can think of some ideas of how to do that. Most importantly, really, a safe harbor really as versatile and easily adaptable to other LSDs or even other situations where you want the blood to secrete a therapeutic protein. So in the next few slides, we'll go through these experiments, which constitute proof of concept studies showing that the human gene-edited HSPCs to express hydrolyase can correct symptoms in multiple organs of MPS I. So we use as we described earlier, we use the RNP AAV system, Cas9 protein single guide, and AAV6. Our guide targets exon 3 of the CCR5 locus, and I'm showing you here a schematic of a generic template. You can see that in this template, you can vary the promoter, you can very easily vary the template, and you can add a selectable or a reporter if that's helpful for you. And so how do you measure target integration in human hematopoietic stem cells? So the first step towards achieving high efficiencies of modification in these cells really starts with having a guide that has good efficiency. For this particular guide, you can see that in cells derived from cord blood, which is shown red here, or peripheral blood shown in blue, you get either 85% or 75% of the cells of the alleles have indels in them. We measure indels using Sanger sequences, and then we have those sequences disconvoluted using freely available software online. I'm giving you an example here on this slide. You can see from the sequences and also from this histogram that the most common indel in this particular system is a single nucleotide insertion. And you can see in the fact plot where we're looking at CCR5 expression that the addition of the guide with the Cas9 or as an RNP decreases the expression of the CCR5 guide. So we are knocking it out when we do this process. You can measure the frequency of target integration by looking at reporter expression. In this case, we're using yellow fluorescent proteins. You can see 3 expression cassettes here. 2 of them have a reporter. One of them doesn't, and one of them is treated by a very strong viral promoter, SFSV, and the other one is a strong constitutive eukaryotic promoter, PGK. You can measure your reporter very easily if it's a fluorescent protein using flow cytometry with fluorescence on the y-axis. You can see the SFSV is stronger than PGK. And you can basically measure how many cells are expressing your protein. So you can see that for cord blood in general, we get a mean average modification of 35% of all the cells, which is not bad. 20-something% when the cells are derived from peripheral blood. Now, if you remove your reporter and your integrated cassette smaller, your efficiencies can go higher. So the efficiencies really depend on the insert size, and you can, if without the reporter, you can look at modification rates that are in the mid 40%. Now you can measure integrations more directly by looking at the look itself. Here I'm showing you the use of droplet digital PCR to accomplish this. There are, of course, other ways to do this. So to do ddPCR, you need a person probe that works as a reference. It could be anywhere else in the genome, but we prefer to place it in the same chromosome. And then you need another probe assay that will read only your integrated version of your allele, and that's sort of very specific to what you want to do. We're looking, we have primers inside and outside the homology arms, and that's how we make sure that we locate integrated DNA only. The assay can be validated, so here we validated it with DNA from a colony that had only one allele modified, so you know the answer, if it works well, has to be 0.5, and you can see that it is. And you can use this to measure the percentage of modified alleles, and also it allows you to confirm that you're dealing with the intended integration and not random integration. Genotyping of colonies derived from single cells, which is something you can do with HSPC, is a great way to quantify cell modification rates when you don't have a reporter for modification. And also it allows you to quantify rates of mono and biallelic integration. So in this particular example, non-integrated alleles are bigger, 1.1 kb, and integrated alleles show a smaller band. So if you have two bands, you're modifying one or two alleles, so monoallelic modification. If you have a single band, you have biallelic modification. So let's talk about how to check for expression of the therapeutic protein. In this particular example, we're looking for expression of a lysosomal enzyme. So here you can see that when you look at the cells, you have some cells that have, based on report expression, YFP high, low, or negative. We are assuming that the YFP high cells are the ones that have the integrated cassette, and we can demonstrate that by showing that these cells are being kept in culture for a really long time, and they will always express that reporter in the enzyme because this cassette is integrated in the DNA. Now, if you compared the modified cells with unmodified cells, you can see that cells that are expressed iduronidase driven by the SFP promoter have several hundredfold more enzyme expression, and some cells that expressed that were modified with the PZK cassettes expressed between 25 and 54 more enzymes. So the enzyme can be measured in lysase, but as I mentioned before, these are normally secreted, so we measure it also in the media. So we are, as we meant to do, we're expressing supraphysiological levels of the enzyme. As I mentioned to you earlier, a central concept of the idea of using HSCT to treat other organs is that the progeny, in particular the monocytes and macrophages, will continue to express stable supraphysiological levels of enzymes, and so this data confirms that that is the case. So you can take your HSPCs and push them in differentiation in vitro with the right cytokines, and even after differentiation, they continue to express lots of these enzymes. Now, the main concern about the use of nuclease-based genome editing platforms for therapeutic purposes is the possibility of creating double-strand DNA breaks at unintended sites, commonly referred to as off-target sites. So there are a variety of ways to measure, this genotoxicity in these cells. Generally, they're divided between bias, meaning bioinformatically predicted, or unbiased approaches. So what I'm showing you here is an example of a bioinformatics guided approach. We use the bioinformatics tool called COSMID to predict off-target activity based on sequence similarity. So it predicted 67 sites, and then you go and sequence them deeply to see if there are modifications in those sites. So when we use a standard Cas9, which is shown here, and wild type, we see 4 sites that have off-target activity, meaning they're above the limit of detection for next-generation sequencing, which is 0.1. They're above the background, and they're repeated in various experiments. So 4 out of 67. You can see that there's very, very low levels of off-target activity, is 0.5. But most importantly, when you switch the Cas9 to a a version that has a mutation that provides it with higher fidelity, those off-target sites go away. Now there are, as I mentioned before, there are more unbiased ways, genome-wide ways to look at this. These tools are being actively developed. I feel like every few months there is a new one. This slide lists some of these tools, though probably not comprehensive, and we classify them between in vitro and in vivo-based approaches. So it's another way to assess off-target activity in cells. And if you're trying to design a long-term, hopefully one-time therapy, you want to target a long-term populated stem cell, and you want to make sure that after the modifications, these cells retain the ability to self-renew and differentiate into multiple lineages. The schematic is supposed to remind you of the structure of the hematopoietic compartment. Remember that there's a bone marrow pluripotent stem cell that can self-renew and also give rise to progenitors, which will give rise to all these different cells in the bone marrow through a series of steps and gradual process of commitment and differentiation. The question of cell renewal is typically addressed by checking if the cells that have undergone an canning graft and serial engraftment studies. If you're using human cells, then you have to use immunocompromised mice. So in this case, we used the not SCID gamma mice, the abbreviated MSG, here, which was sent to cells that were selected based on reported expression. And I'm looking at showing you data from primary engraftment studies. So after 16 weeks, you look to see if there are human cells in bone marrow and other organs. You can see that there are cells there, and the cells that are engrafted are, if you engrafted YSP positive cells, at the end they're YSP positive, confirming that these cells were in fact modified. You can also notice that there is a decrease in engraftment potential if the cells have undergone genome editing. We think that this is because there are fewer repopulating clones in that population, and this is based on observations that HR is less efficient in cells that are more quiescent, which are really your true stem cells. So also in the mice, you would be able to look at, whether you're producing B cells, T cells, and myeloid cells. I'm not showing you that data, but this experiment is also allowed to show that there is multi-lineage differentiation. That can tell you that these cells differentiate normally. Now, perhaps the most important question is to determine the efficacy of your cells, and the standard way of doing this is using animal models. Starting with human cells is great because they're ultimately your target cells, but this adds significant limitation for testing efficacy in animal models, as you have to contend with the potential of a rejection of the cells. So to get around this, for this particular example, we had to make a new mouse model of the disease that was capable of engrafting human cells. So we used CRISPR to knock in a severe human mutation into NSG mouse embryos. And to make a long story short, these animals replicate the disease, fortunately for us, the same as predicted from patients, and it had been done previously for immunocompetent mice. You can see that they lack enzymatic activity, they accumulate gags, and they have the skeletal and visual manifestations that you would expect. So when you transplant our human modified cells in bulk, these were cells, for example, that didn't have a selectable marker, you can see that there are positive impacts on the biochemical marker. So on the right, you can see that enzymes are constituted, so you're comparing red dots to blue dots primarily. In the liver and spleen, you get supra, you get more than normal enzymatic activity, and this results in an overall decrease in GAG storage, which is measured in the urinary excretion of GAGs. In some tissues like liver and spleen, the GAG storage is completely reversed. In the brain, we don't see the same types of differences, and we'll come back to this. Consistent with the biochemistry, the transplantation will improve phenotypic abnormalities. So this results in an improvement in a skeletal phenotype, and whether you transplant the cells in bulk or sorted, especially in the central nervous system, we see improvement in neurobehavioral parameters. And this graph you really want to compare. So the red bars with the red lines to the blue lines. And you can see that transplanted mice have improved spontaneous locomotion activity, they improve short-term memory and digging behavior. So it is positively affecting the central nervous system. And so just wanted to summarize here that, so we have human HSPCs can be in here with good efficiencies. I showed you an example of using CGR5 safe harbor locus, particularly we're trying to observe these large expression cassettes. It works really pretty well. We've shown you that the cells remain stem cells after modification, that we preserve this long-term repopulation, multi-use differential potential, and actually these cells are capable of improving disease phenotype in the mouse and non-hematological disease phenotypes, skeletal and brain, as well as biochemical phenotypes in liver and spleen. So we describe this as a potential platform for lysosomal enzymes, but you can be more imaginative and say, well, this actually is a platform for secretable therapeutic proteins. So technically, we could try to think about using this for other lysosomal enzymes. There's more than 15 of these diseases. But I wanted to give you another example. So in the example that I described, we use constitutive promoters that really engage every cell, including the hematopoietic stem cell, for enzyme expression. And we knew that we could do that because it didn't really affect the enzyme itself. It doesn't affect the hematopoietic stem cell, the progenitors, but this was a way to maximize protein expression. There might be cases where this is not what you want to do. You might want to restrict it to certain types of cells. So here I'm showing an example of another enzyme called glucocerebrosidase, in which that expressing in early compartments could affect engraftment. So we restricted expression to the cells that we really care about, which is the monocytes and macrophages, especially in the disease that's that is the result of glucocerebrosidase absence, which is Gaucher disease. So you can see that this safe harbor strategy can be quite flexible. You have one therapy for all your patients, all their mutations, and in addition, you can use this tissue and lineage-specific promoters to make the system even more customizable. So I think now it's time to wrap up. Here, what I think are the most important take-home messages. Therapeutic and HGA can be used to decrease or increase expression of target proteins. Therapeutic HDR allows you for precise genetic modification and the ability to synthesize these new coding sequences and regulatory sequences, which really opens up more therapeutic options. The safe harbor approach is a flexible platform for the expression of therapeutic proteins. Hopefully, we have shown you that hematopoietic system can deliver these therapeutic proteins to other organs. The primary effector cells we know for the tissue delivery are the monocytes and macrophages, and such strategy is really most applicable to secreted proteins. And now I would like to open it up for questions.
Thank you, Dr. Gomez-Ospina, for your informative presentation. We will now start the live Q&A portion of our webinar. Now, to our audience, if you have a question you would like to ask, if you haven't already done it, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we will answer as many questions as we have time for. Now, let's get started. Okay, Dr. Gomez-Ospina, let's start with this first question. Can you talk about how you narrowed down your culture conditions for editing the CD34 positive cells?
Well, that's a great question because we have been using culture conditions that have been reported in the literature for a while, and to be honest, a lot of the people who are doing this haven't really thought very carefully about, whether we're really using the best conditions possible. So, there are out there different options for that. So the bottom line is we know very little about how to culture the cells outside of the body, and so people are working on different types of cultures, whether to keep the media static or change it continuously. There's a set of cytokines that everybody uses, but nobody really understands the doses. We know that we're certainly adding too much of certain cytokines with the idea of hoping to expand these cells outside of the body, they are actually a bunch of new small molecules and even cytokines that are being added to these cultures. So to make a sort of a long answer short, we've done very little optimization, and I think there is a lot to be done. So we're basically using culture conditions that have been described for a while.
Thank you. Now our next question. What are some barriers you see in moving CRISPR-based cell therapies from the lab to the clinic?
So that's an incredibly complex question, and we could talk for a long time about this. I think a big barrier for all kinds of therapies, whether they're CRISPR-based or cell-based or gene therapy-based, the biggest barrier really is financial incentives. For rare diseases, there were, not that long ago, we had plenty of diseases for which there were no experimental therapies. So right now, they're actually competing therapies, which a lot of them, are viral-based, and some are easier to deliver, and some have been in development for a longer time. So it's sometimes hard to find money to work on a therapy because you have all these other therapies. But to be more precise, there is the challenges that are sort of at the level of preclinical research. So when you're doing your research in animals, one is assessing efficacy, meaning, there's this, are you really using a suitable animal model? And that really applies to almost any therapy, but this is particularly tricky when you're trying to engineer human cells. And so you have very little options in terms of testing them in an animal model. Perhaps more importantly, really, is the safety aspect of assessing the specificity of these genome editing technologies. This is a space that's kind of changing very quickly in terms of tools that are available, and it's also kind of unclear what the regulatory agencies are really requesting, that you really have to show them in order for them to be convinced that your approach it's really safe. And the safety is not just about the genotoxicity of making double-stranded breaks where you don't want them, but it also, a very important problem for everyone that is trying to replace expression of genes or proteins that were not in the body before, is the immunogenicity issue that either from your vectors, but very specifically also from for our CRISPR editing tools. A lot of challenges.
Thank you. Will RNP positive AAV6 EP affect HSPC viability?
So we know that AAV alone will decrease the clonogenicity of the HSPC. So if AAV itself has an effect on clonogenicity. It's true, and so we are subjecting these cells to a complex process. Also, electroporation also triggers a variety of activates a bunch of signaling pathways as well. It does.
Thank you. Our next question. Have you tested the effects of static versus continuous flow bioreactor type cultures?
We haven't. We would like to. I think that that's an important question. What happens with these cultures, particularly progenitor cells, can be expressing cytokines that may be inducing differentiation or compromising stemness. So I think that it's very likely that better sort of continuous flow systems would have positive effects on maintaining stemness in these cultures.
Our next question. So the half-life of modified stem cells is how long, and how do they regenerate?
So if we're talking about hematopoietic stem cell and progenitor cells, the half-life really depends on where you're testing them, right? We've kept them in vitro for a really long time, months. In the mice, you could do these serial transplantation experiments that are really about trying to capture those true stem cells that are capable of self-renewing for a long time. So you do a first experiment that varies between 4 and 5 months. You take the cells and you put them in another mice. You wait another 4 and 5 months, and you can do it again. So these serial transplantation experiments, we've done them for 40 months, but some people even do an additional random transplantation. In humans, we expect these cells should last forever if they're true stem cells, and that's the whole point of trying to really do the best that you can to confirm that these cells are stem cells.
Now, Dr. Gomez-Ospina, have you had any issues acquiring a high enough titer of AAV6 to be used as a donor template?
Enough titer? No, I mean, we have conditions where there's definitely too much AAV on the cells that it becomes. So usually when we start an experiment and we try to design a new donor template, we do a titration experiment where we kind of find the point where we maximize editing efficiency and sort of kind of find the sweet spot with the effects on viability. And so what I would say is generally getting enough titer is not the problem. There are a lot of other issues with how much AAV will actually, get liberated from the endosomes and how that template will actually be used for HR. So every locus is different, and we find that we really haven't found a really good way to predict which ones will have really good efficiencies to do the HDR part of the repair that requires a template. And so we don't really know what goes into that.
Thank you. Now we have some great questions coming in, and we may not have time to get to all of them. So I do want to remind our audience that those questions we do not have time to get to today, and those that are submitted during our on-demand period will be answered via the email address you provided at the time of registration. Okay, but let's keep trying to go through our QA here. Dr. Gomez-Ospina, can you share with us what the potential side effects of gene therapy using gene editing are?
So I guess the one issue that everyone talks about is the possibility of creating a change somewhere in the genome that you didn't intend. Usually most people are worried about, disrupting, activating oncogenes or disrupting tumor suppressor genes that could lead to tumorigenicity in the future. This is particularly important in HSPCs, or the protocol that I've described to you, because you really are modifying a cell population. And so within that within those millions of cells that you potentially transplant into the patient, there could be a few clones that could have those changes. So so that is sort of theoretically a real possibility. Let me remind you that people have been using lentiviruses, which integrate randomly or sort of randomly into genomes, and those have actually made it into people. So, there are always risks and benefits to be weighted. I think the other issue that we haven't paid enough attention to, as I mentioned earlier, is the issue of immunogenicity. So the nice thing about doing these things ex vivo is that if you let's say there's data now showing that these Cas9 proteins come from bacteria that are very common in people, and we have antibodies and we have T cells against them. So doing this ex vivo can probably reduce the likelihood that you're going to respond to that. Also could be a potential problem if you're trying to use this in vivo. But also there could be immunogenicity to the protein itself. So if you're trying to replace a protein that a patient with a genetic disease didn't have, they may recognize it as foreign. So it's something else to think about. Those are two potential pitfalls. I'm sure there are more.
Very good. Thank you. Now, Manuel Leonetti has a paper in bioRxiv that showed that HDR may not be as accurate as it may be, showing that parts of large inserts can be mutated in different types of donors like plasmids, SSODNs, and PCR products. Have you noticed anything like that with AAV6 donor?
I haven't looked too much into that, to be honest, but I know that other people have noticed those changes. They're rare. I mean, the majority of the times you get the appropriate change, but yeah, you can have errors. And it's interesting because different types of cells have different repair mechanisms. When you do this kind of work in embryonic stem cells or even embryos, you get surprising, very different types of repair. So yeah, and I think that, the tools that we have so far, which we're just kind of focused on the one little area that we're trying to change, are really not allowing us to see the entirety of the changes that we're making.
Now, do you think there is an ideal level of activity for the enzyme from the modified cells, or should we restrict this to avoid other off-target effects?
So for each therapeutic protein or enzyme, really, it varies, and I think you need to sort of kind of get an idea of what that is from the literature or experimentally. So some levels. So for the example that I showed you with this enzyme hyaluronidase, we knew that high levels were okay because people had overexpressed it hugely in mouse cells, and there was no problems with engraftment and differentiation. There are other enzymes where you probably want to restrict the activity. So as I showed you, using different promoters allows you to kind of play with that a little bit. So that's in terms of like regulating the levels of the therapeutic protein, at the levels of the protein themselves. In terms of regulating, the actual editing events, that's an interesting question. We're not at the point where we're getting so much editing that we are wanting to like have less of it. I think for every if you're trying to design if you're starting an approach with a genome with a nuclease-based platform, what you need to do really early on is especially if you're using CRISPR-Cas9, is to focus on find a good guide that gives you good efficiencies and characterize the off-target effects of that guide to the best of your ability as early on in the process, so you don't later find out that that particular guy has many off-target sites, and so it's something you should try to do early on.
Thank you. We're going to try to squeeze in just a few more questions. What approach, in vivo versus ex vivo, would be suitable for correcting single base mutation in sickle cell disease in human subjects?
So I'm most aware of ex vivo approaches to correct sickle cell disease in humans. Using exactly the same protocol that I described, a guide to the HBB gene, Cas9 protein, actually now using higher fidelity versions of the Cas9 and using AAV6 to deliver the donor template. So that's already in development. I mean, there are different variations of this approach, and that people may not be using AAV6, and people could be using single-stranded DNA oligos. And so I'm only aware of the ex vivo approaches. Theoretically, you could try to do it in vivo. I'm not sure how those efficiencies would compare. But again, the nice thing about doing this ex vivo is it allows you to kind of really characterize some of these concerning issues that we're talking about, like, off-target activity, et cetera, and correction before you actually deliver the product to the patient.
Got it. Our next question, what about the delivery system effect?
Well, I think we started talking about that a little bit earlier. So, we are putting the cells through a lot. It involves, these are HSPC, so some of these progenitors are sort of they are about to be immune cells. Some of them have pretty aggressive immune recognition systems, and so we're exposing them to Cas9, we're exposing them to an RNA, we're exposing them to electroporation, and we're exposing them to AAV. So as far as we know, so you can measure gene expression changes. In the past, when people started doing these experiments, they were using either plasmids or naked RNA, or the Cas9 was delivered as an mRNA or as a plasmid. And what we know is that delivering as protein and a modified guide really decreases a lot of those effects. But there are some, and then there's, like, as we discussed earlier, the AAV has effects on the HSPCs as well.
Thank you. Can we use the HSPCs as a therapy for inflammation of neurodegenerative disorder?
I think so. I think I saw an encouraging paper recently. I don't remember. It was very recent, but I think, I think there's good evidence to think that would be possible.
Thank you. And Dr. Gomez-Ospina, we're going to have to wrap with this final question here. How can we make sure that the cells that we save after re-injection into patients, especially because these cells were in vitro expanded?
How can we make sure that the cells are saved?
Are saved, yes, after re-injection.
So, I mean, if I understand this question correctly, so normally, these earlier trials will probably require that you have a backup sample of the patient's cells that are unmodified to be able to rescue in case, the cells don't work. I'm not sure if that's where the question is going. But yeah, so, for patients specifically, there'll probably be a requirement to have a backup of unmodified cells that you can use in case your therapy doesn't work.
Very good. And if we didn't answer that question correctly, please feel free to submit that question again, Eamon, and we'll try to answer your question more correctly. But I want to thank all of our attendees who submitted their questions. We had a great Q&A session today. But I really want to thank Dr. Gomez-Ospina for her time today and her important research. We'd also like to thank LabRoots and our sponsor, STEMCELL Technologies, for underwriting today's educational webcast. As a reminder to our audience, those questions we did not have time for today, and those submitted during our on-demand period will be addressed via the contact information you provided at the time of registration. Now, this webcast can be viewed on demand, and LabRoots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now, and we hope to see you again soon. Have a great day.